What if I told you there's an AI that can see chemistry before it happens? Not predict the weather, not generate cat pictures, but actually watch molecules dance together in ways that took nature 4 billion years to figure out. In May 2024, DeepMind quietly released something that pharmaceutical executives are losing sleep over. It's called AlphaFold 3, and it just did something that should have taken humanity another 50 years. Here's what I mean. Right now, somewhere in a university basement, a graduate student is spending their 4th year trying to figure out the shape of a single protein. They're using X-ray crystallography, a technique that involves growing crystals, blasting them with radiation, and praying the math works out. Cost? Anywhere from $50,000 to $500,000. Timeline? Months. Sometimes years. Sometimes never. AlphaFold 3 does it in minutes. But here's the thing that nobody's really explaining properly. AlphaFold 3 isn't just faster. It's playing an entirely different game. Think of it like this. Imagine you're trying to understand how a Lego set works, but you can only look at the bricks. That was AlphaFold 2. Revolutionary? Absolutely. Over 2 million researchers in 190 countries used it. It predicted the shape of 200 million proteins. That's 1,000 times more than all the experimental structures humanity had ever determined combined. But knowing the shape of a Lego brick doesn't tell you what you can build with it. It doesn't tell you how bricks snap together. It doesn't tell you what happens when you add other materials, maybe some Duplo blocks, some Technic pieces, some completely custom parts. AlphaFold 3 sees the whole picture. It predicts proteins, yes, but also DNA, RNA, small molecules, drugs, metal ions, and this is the crucial part, how all of these interact with each other. This is what scientists call the molecular machinery of life, and AlphaFold 3 can simulate it before it happens. Let me give you a number that should make you pause. For protein-ligand interactions, that's proteins binding to potential drugs, AlphaFold 3 doubled the prediction accuracy compared to the best existing methods. Doubled. In a field where a 5% improvement gets you published in Nature, they achieved 100% better performance. So why should you care? Why should anyone outside a research lab care about molecular predictions? Because this is how drugs get made. Or more accurately, this is how drugs fail to get made. The pharmaceutical industry has a dirty secret. It takes on average 12 years and $2.6 billion to bring a single drug to market. And here's the kicker. 90% of drugs that enter clinical trials fail. 9 out of 10. That's not just wasted money. That's wasted time. That's patients who didn't get treatments that might have worked. A huge portion of those failures happen because scientists misunderstand how a drug molecule will interact with its target protein. They build what they think is the perfect key for a lock, but when they actually test it, the key doesn't fit. Or worse, it fits somewhere else and causes side effects. Now imagine if you could see, before you synthesized a single molecule, before you ran a single experiment on a mouse, before you spent $1 on clinical trials, imagine if you could see exactly how that drug would bind to its target. That's what AlphaFold 3 offers. And Isomorphic Labs, DeepMind's drug discovery spin-off, is already betting billions on it. They've signed deals worth over $3 billion with pharmaceutical giants Eli Lilly and Novartis. These aren't speculative investments. These are hard-nosed pharma companies saying, this technology is real enough to stake our pipeline on. But here's where things get really interesting. Let me tell you how AlphaFold 3 actually works, because the architecture is genuinely wild. You know those AI image generators? Midjourney, DALL-E, Stable Diffusion, the ones that create photorealistic images from text prompts? They all use something called diffusion models. You start with pure noise, random static, and gradually remove the noise until a coherent image emerges. AlphaFold 3 uses the same principle, but for molecules. Think about that for a second. The AI doesn't build a protein step by step, amino acid by amino acid. It starts with a cloud of random atomic positions, just noise in 3D space, and gradually refines that noise until the correct structure emerges. It's like the molecule was always there, hidden in the static, and the AI just finds it. This diffusion approach is why AlphaFold 3 can handle so many different molecule types. Previous versions had specific architectures for proteins. You put in a protein sequence, you get out a protein structure, but real biology isn't that clean. In your cells right now, proteins are grabbing onto DNA, reading RNA, getting modified by sugar molecules called glycans, picking up metal ions like zinc and magnesium, and sometimes all of this is happening simultaneously. One protein might have 3 different partners at different times, and the shape it takes depends on who it's dancing with. AlphaFold 3 sees all of this. You can input a protein sequence alongside a DNA sequence, add a potential drug molecule, throw in some chemical modifications, and it will predict how the whole system fits together. Let me give you a specific example that matters for human health. Antibodies. Antibodies are your immune system's guided missiles. They're proteins that recognize and bind to specific targets, usually foreign invaders like viruses or bacteria. But engineering antibodies as drugs has been challenging. You have to find an antibody that binds tightly to your target, doesn't bind to things it shouldn't, and actually does something useful when it connects. This involves testing thousands, sometimes millions of candidates. It's expensive, slow, and a lot of it is educated guessing. AlphaFold 3 predicts antibody-antigen interactions with unprecedented accuracy. You can now computationally screen antibody designs before ever synthesizing them. This isn't theoretical. It's already being applied to vaccine development and cancer immunotherapy. And speaking of vaccines, remember those mRNA vaccines that saved millions of lives during COVID? Those work by delivering genetic instructions, RNA, into your cells. But RNA is notoriously finicky. It folds into complex shapes that determine how well it works, how stable it is, how efficiently cells can read it. AlphaFold 3 can predict RNA structures and protein-RNA interactions. This opens the door to designing better mRNA therapeutics, not just for vaccines but for gene therapy, for treating genetic diseases at their source. Now let me pull back and explain why I said this should have taken 50 years. The problem of predicting how molecules interact has been called intractable by serious scientists. Not just hard, fundamentally unsolvable with existing approaches. The number of possible ways molecules can fit together is astronomical. A single protein can have millions of possible shapes. When you add multiple molecules interacting, the combinations explode beyond any computer's ability to search through them. Traditional computational methods, molecular dynamic simulations, docking algorithms, they work, sort of, but they're slow and often inaccurate. The joke in the field is that computational predictions are useful for generating hypotheses, but you'd never trust them enough to skip experiments. AlphaFold 3 changes that calculus. Its predictions are accurate enough that researchers are starting to trust them for actual decision-making. Not replacing experiments entirely, we're not there yet, but reducing the number of experiments needed by orders of magnitude. But I need to tell you about a controversy, because this isn't all good news. When DeepMind released AlphaFold 2, they eventually made everything public. The code, the trained model weights, everything. Scientists could run it on their own computers, modify it, build on it. This open approach turbocharged research. Thousands of papers, countless discoveries. The scientific community embraced it. AlphaFold 3? Different story. DeepMind initially released AlphaFold 3 only through a limited web server. You could use it, but only through their interface, with restrictions on how many predictions you could run. The model weights? Not released. The full code? Not available. This sparked significant pushback from the scientific community. Some researchers called it a step backward for open science. Others pointed out potential conflicts of interest. Isomorphic Labs, remember, is using this technology for commercial drug discovery. If competitors can't access the full model, that's a substantial business advantage. Now, DeepMind has said they're committed to openness and are working on releasing more. They cited safety concerns and the need for responsible deployment. But the tension is real. We're entering an era where the most powerful scientific tools might be controlled by a handful of companies. This matters because science thrives on replication and iteration. When only one group can run the best tools, when others can't check their work or build on it, that's a fundamental shift in how knowledge advances. So, where does this leave us? What does the next 5 to 10 years actually look like? Here's my honest assessment. AlphaFold 3 is not a magic wand. It has limitations. Its predictions, while dramatically better than previous methods, still have error rates. Some molecule types are harder to predict than others. The model can confidently output incorrect structures. It doesn't know what it doesn't know. Experimental validation is still essential. You still need wet lab work. You still need clinical trials. The entire drug development apparatus doesn't disappear overnight. But, and this is a critical but, the bottleneck shifts. Previously, just understanding the basic shape of a target protein could be a multi-year project. Now, that's a query to a database. Previously, generating hypotheses about how a drug might bind was expensive speculation. Now, you can computationally screen thousands of compounds before touching a pipette. The drug discovery process compresses, not to zero, but meaningfully. Where it used to take 12 years, maybe now it takes 8. Where it used to cost $2.6 billion, maybe now it's $1.5 billion. Those are rough numbers, but the direction is clear. And more importantly, drugs that were previously undruggable targets, proteins that were too hard to study, too complicated to design compounds for, those become accessible. There's another effect that I think people underestimate: democratization. When the AlphaFold database went public with its 200 million protein predictions, it was like giving every university, every small biotech, every researcher in developing countries access to what previously only the most well-funded labs could generate. Structural biology used to require million-dollar equipment and years of training. Now a student in Lagos or Bangalore can access the same molecular information as a professor at Harvard. Yes, there are concerns about AlphaFold 3's restricted release, but even the limited web server gives researchers unprecedented capability. And as competition heats up, Meta, other companies are working on similar tools, the pressure toward openness will likely increase. Let me leave you with something concrete. A list of diseases where AlphaFold 3's capabilities are already being applied. Cancer. Understanding how oncogenes work. Designing targeted therapies. Alzheimer's and Parkinson's. Studying protein misfolding. Finding ways to intervene. Malaria and tuberculosis. Developing drugs for neglected tropical diseases. Antibiotic resistance. Designing new antibiotics when bacteria evolve to defeat old ones. Genetic disorders. Understanding how mutations cause disease. Developing gene therapies. This isn't hype. These are active research programs using AlphaFold predictions right now. The fundamental shift here is that biology is becoming an information science. For most of history, understanding life meant observing it, looking through microscopes, running experiments, watching what happens. The molecular world was dark, accessible only through indirect and expensive measurements. AlphaFold 3 turns on the lights. Not completely, there's still darkness at the edges, but enough that we can navigate in ways that were impossible before. The AI doesn't replace chemists or biologists. It gives them X-ray vision. It lets them see what was invisible, simulate what was unsimulable, try things computationally that would take lifetimes to try experimentally. So here's the bottom line. AlphaFold 3 is not another AI hype story. It's not a chatbot. It's not generating art or writing essays. It's doing something much more specific and arguably much more important. It's mapping the chemical terrain where life happens. Every drug you've ever taken works by interacting with molecules in your body. Every disease is, at some level, molecular machinery gone wrong. Understanding these interactions is the foundation of medicine. We just got dramatically better at understanding them. Not because we had a brilliant insight, but because we built a machine that can see what we couldn't. If you found this breakdown useful, hit subscribe because we go deep on how technology actually changes the world, not the hype, the real mechanisms. Next week, we're looking at the race to build the next generation of AI chips, and why Taiwan keeps the entire tech industry up at night. I'll see you there.